Unsaturated fatty acids are mainly cis isomers in our body and are also oxidized by beta oxidation. Beta oxidation of unsaturated fatty acids are also continued within the mitochondria until either a delta 3 cis acyl-CoA or a delta 4 cis acyl-CoA is formed, depending upon the position of the double bond. For example, if it is a oleic acid, after 3 cycles of beta oxidation, delta 3 cis enoyl-CoA will be formed, which will be isomerized to delta 2 trans enoyl-CoA by a cis trans isomerase and then again undergo beta oxidation. If it is a linoleic acid, after 4 cycles of beta oxidation, delta 4 cis enoyl-CoA will be formed, which by acyl-CoA dehydrogenase will be oxidized to delta 2 trans delta 4 cis dienoyl-CoA and then will be reduced by dienoyl-CoA reductase to delta 3 trans enoyl-CoA and then will be isomerized to delta 2 trans enoyl-CoA by delta 3 cis delta 2 trans enoyl-CoA isomerase. This is a positional shift isomerism, and then delta 2 trans enoyl-CoA again undergo beta oxidation. Hypoglycin, found in unripe ackee fruits, is an amino acid which is metabolized in our body by deamination and oxidative decarboxylation to methylene cyclopropyl acetyl-CoA, which inhibits acyl-CoA dehydrogenase during fatty acid oxidation, or it inactivates carnitine palmitoyl transferase 1 or thiolases during beta oxidation. This is characterized by gastrointestinal illness, vomiting, hypoglycemia, and CNS depression. Severe hypoglycemia and seizures ultimately leading to coma and death, mainly in case of malnourished children. Ackee tree mainly found in West Africa and is the national fruit of Jamaica. It is delicious and staple food of Jamaica. Unripe ackee fruit, when it is shelled, it contains hypoglycin and produces toxicity, which is commonly known as Jamaican vomiting sickness. Similar thing happened in India last year. About 100 children of Muzaffarpur, Bihar died by lychee fruit. Very long chain fatty acids, that is C20, C22, C24 fatty acids diffuses into the peroxisome and thioesterified by very long chain fatty acid thiokinase to very long chain acyl-CoA, which is then oxidized by aerobic dehydrogenase acyl-CoA oxidase to form delta 2 trans enoyl-CoA. This step is different from mitochondrial beta oxidation. In this reaction, FAD is reduced to FADH2, which donates its hydrogen directly to molecular oxygen, forms FAD and H2O2. A catalase converts hydrogen peroxide to water and molecular oxygen. Then, like beta oxidation within the mitochondria, delta 2 trans enoyl-CoA hydratase catalyzes the hydration to produce 3-hydroxyacyl-CoA, which is again dehydrogenated by 3-hydroxyacyl-CoA dehydrogenase to form 3-ketoacyl-CoA. Here NAD+ is also act as electron acceptor and NADH is produced. Then thiolase cleaves the 3-ketoacyl-CoA into acetyl-CoA and a 2-carbon shorter acyl-CoA is produced. Peroxisomal thiolase has substrate specificity longer than C8, so peroxisome could not metabolize octanyl-CoA and is transferred to mitochondria for further beta oxidation. Zellweger syndrome is a rare congenital disorder, typically appears in newborn, described by Dr. Hans Zellweger in 1964. It is a paroxysmal disorder that occurs when paroxysms are missing or do not function correctly. Accumulation of long-chain fatty acids, hydrogen peroxide in cells may cause the damage. Children with this syndrome have poor muscle tone, skeletal abnormalities, distinctive facial features, hearing loss, vision loss, etc. Beta oxidation is the main metabolic pathway for fatty acid oxidation, but in our body alpha and omega oxidation of fatty acids also take place. In brain, alpha hydroxylase catalyzes alpha oxidation of very long-chain fatty acids for incorporation into cerebrosides and sulfatides. For example, lignoceric acid, C24 carbon fatty acid, by alpha oxidation forms 2-hydroxy-lignoceric acid or cerebronic acid, which is incorporated into cerebrosides called cerebrons. Some hydroxy fatty acids produced by alpha oxidation may be oxidatively decarboxylated to produce odd-chain fatty acids for incorporation into brain sphingolipids. Phytanic acid produced by dietary phytol is oxidized by phytanate alpha oxidase and then by oxidative decarboxylation produces odd-chain fatty acid pristanic acid, which undergoes beta oxidation and produces acetyl-CoA and propionyl-CoA. Genetic deficiency of phytanate alpha hydroxylase may cause accumulation of phytanates in tissues which produce different neurological symptoms. This disease is known as Repsum disease. Omega oxidation is a very minor pathway by the enzyme omega hydroxylase, a enzyme of microsomal cytochrome P450 oxygenase system which is present in smooth endoplasmic reticulum of liver. Omega hydroxylase hydroxylates omega methyl group of the fatty acid to hydroxymethyl group and after oxidation produces dicarboxylic acid which can undergo beta oxidation from the both end of the fatty acid CoA and produces acetyl CoA as well as C6 and C8 dicarboxylic acids, adipic acid and sucraric acid respectively. Inherited deficiency of mitochondrial mid-chain acyl-CoA dehydrogenase may cause accumulation of mid-chain fatty acids and are oxidized by omega oxidation and produces these dicarboxylic acids and are found in urine, which is known as dicarboxylic aciduria. If the chain length of a even carbon fatty acid is n, for example, palmitic acid, a 16 carbon fatty acid will produce 8 units of acetyl coenzyme A by 7 cycles of beta oxidation. If the fatty acid is odd chain fatty acid and chain length is O, then O-3 by 2 times beta oxidation cycle will be repeated to produce O-3 by 2 number of acetyl CoA and 1 3 carbon unit propionyl CoA. For example, heptadecanoyl CoA, a 17 carbon fatty acid, will produce 7 units of acetyl CoA and 1 propionyl CoA by 7 cycles of beta oxidation. Propionyl CoA does not undergo beta oxidation and is carboxylated by propionyl CoA carboxylase to methylmalonyl-CoA, which is then epimerized to succinyl-CoA by methylmalonyl mutase. Succinyl-CoA is an intermediate of TCA cycle. Vitamin B12 acts as a cofactor of methylmalonyl-CoA mutase, so its deficiency in pernicious anemia patient excrete large amount of methylmalonic acid in urine.